So, welcome to the third session of our global webinar to celebrate fungi and explore fungal diversity, interactions and their uses. And we've already had several sessions today. The first session covered talks from Asia and Australasia, the second one from Africa and Europe, and now this session covers talks from the Americas. In the past, we've managed to have talks from Antarctica, and indeed Bob Blanchette, who you can see as one of the speakers today, has talked to us about Antarctica, but we haven't got any from that continent today. So we have 4 talks now from Matt, Patricia, Robert and Michael, and they will give their talks one after another with no questions in between. We'll take the questions and have a good discussion at the end of that first 2 hours. So please put your questions in the chat as you think about them, and then I will note them down and ask those questions of the speakers at the end. So I think at this point, welcome to all of you, welcome to the listeners and to our 4 speakers, whom we very much look forward to hearing. And I will welcome Matt Kasson to give the first presentation. Matt is an associate professor of mycology and forest pathology at the University of West Virginia, USA, and he researches into things like fungal diseases, fungal arthropod interactions, biocontrol of invasive plants, pests, and pathogens. So if you could share your screen, Matt, that would be great. And Matt's going to tell us about interactions between fungi and invertebrates from Arthrophaga to Zoopthora, a world tour of the diversity of fungi that consume arthropods.
Thank you, Lynn, and thanks to the British Mycological Society for having me. I'm really happy to be part of the UK Fungus Day 2024. I'll go ahead and share my screen here. And I'm going to go into presentation mode here. All right, it looks like it might be displaying a reverse. Quickly swap that. Could someone just confirm whether or not you're seeing my display notes or the...
Yes, yes. You need to swap the display.
All right, there we go.
That's it. Thank you.
Okay, thanks. Well, today I'm going to be talking about my work on fungus arthropod interactions, including Arthrophaga to Zoopthora, a world tour of diversity of fungi that consume arthropods. Here we can see a nice collection of diverse arthropods, showcasing their fungal infections, everything from Arthrophaga and Mesospora to Cordyceps and Ophiocordyceps, to Acanthamyces and Pandora. I hope to take you kind of on a world tour of the fungi that are involved in infecting and consuming arthropods, and tell you a little bit about the ones that I work on specifically here in the United States. So where do these insect-destroying fungi fall? I think we need to look at a modern-day phylogeny or family tree of kingdom fungi, and this was one published somewhat recently in Current Biology in 2021. Now, if we look at this fungal family tree, we can see 2 red arrows. These are not by any means the only insect colonizing fungi. In fact, there's been multiple independent groups that evolved to infect and consume arthropods. This includes arachnids, myriapods, insects. Of course, we often talk about insects because they are so diverse and there's so many of them. But there are plenty of non-insect arthropods that succumb to fungal infections. So today I'm going to be highlighting the Entomophthalales, which are here in this red arrow, as well as the Hypocreales, which are ascomycota. Now, there are other ectoparasites like the Lobulobineales, and these are important, but they'll be excluded for this talk given the time constraints today. Now, entomopathogenic fungi are interesting because they've evolved to consume insects, but some of them have developed interesting ways to modify the behavior of their hosts. And in 2021 or 2020, we published a PLOS pathogens paper where we kind of showcased the different lifestyles of these entomopathogens. You could see some modify the host behavior like Strongwellsia and Infuria and Ophiocordyceps and Mesospora. They'll all infect and modify the behavior of the hosts, but some do it while the host is still alive. Others will manipulate the host to coerce that host to climb to a high spot, as is the case of summit disease, die, and then sporulate and rupture out of the cadaver post-mortem. So there are different means by which they can manipulate their host before and after death to maximize spore dispersal. There's some really important, medically important compounds that are produced by entomopathogenic fungi. Among them, cyclosporin is one you've probably heard of. That's an immunosuppressant drug, a very important immunosuppressant drug that allows organ transplants to take place. Cordycepin is another one produced by cordyceps. Cathinone and psilocybin are ones you may not be familiar with, although psilocybin is probably something you recognize from the magic mushroom group. And we'll talk a little bit about that here as we move forward. And then, of course, ergot alkaloids like LAH and ergine and ergonovine. These are all really important compounds, and in some ways are used to manipulate their hosts. But they're also important because they have medical utility in human medicine and human pharmaceuticals. And some of these compounds are used for migraine treatment, and like I said, for organ transplant. So really important compounds are coming out of the interactions between fungi and their arthropod hosts. So when we think about the Hypocreales, the order which contains a lot of the insect colonizing, insect infecting fungi, we often think about the cordyceps term. And this is a term that's kind of outdated, or at least inclusive of several different families of fungi. This is a beautiful tree that you're not meant to read the details, but really look at the color coding here. And this was produced by my colleague Joao, that shows that cordyceps fungi, as a kind of generic term, encompasses the Ophiocordyceptaceae, the Claviceptaceae, and the Cordyceptaceae. And these are all really important families. When we think about the zombie ant fungi, of course, we're thinking about Ophiocordyceptaceae. When we're thinking about true Cordyceps militaris, we're thinking about the Cordyceptaceae. When we're thinking about Metarhizium, which is another important entomopathogen, we're thinking about the Claviceptaceae. So I'm going to highlight a few of those, but just to kind of show you that this term cordyceps, as it's used by the media and by Hollywood, actually encompasses quite a bit of diversity, covering a span of at least 3 fungal families. Now, the where of these fungi is an important question and an important point. We can use contemporary observations that are reported in things like iNaturalist to map where the contemporary Cordyceptaceae are. Here is a map here. That doesn't mean that areas that you don't see these orange squares are areas devoid of Cordyceptaceae. It just means that there's a lot of contemporary observations from those areas, with the darker squares indicating higher numbers of observations. Certainly, if an app was developed and a platform was developed in places like the United States, use there is going to be greater. Similarly, we see a lot of users in Europe, in parts of South America, but very little activity in Africa. In Southeast Asia, we're seeing use, as well as Australia. But again, just because observations are lacking from these areas, it could just highlight areas we should focus on in the future to look for contemporary examples of Cordyceptaceae in Northern Africa. The Cordyceptaceae includes greater than 25 genera, and this includes the Acanthamyces. This includes Beauveria, Cordyceps, Gibellua, Beauveria bassiana, as seen here on this periodical cicada, is used on a lot of other insects as a biological control agent. We can easily culture it. We can easily apply it. It's quite pervasive once it's in the environment, and it's pretty cosmopolitan. You can find Beauveria bassiana or closely related fungi on most continents. Cordyceps militaris is also an important example. We can see that in the picture here, as well as here. We call this the caterpillar fungus, and this is among the most well-known example in the Cordyceptaceae. Of course, things like Acanthamyces and Beauveria are also important, but we often think about the cordyceps themselves and their utility for medical use. I don't specifically work on the Cordyceptaceae, although I'm starting to find interest in them. Around where I live, I find myself in awe, as you can see here in the picture, of moths that are parasitized by these Acanthamyces. Acanthamyces includes what we would formerly call Licanicilium. And for a long time, we thought these were kind of just asexual morphs that we were seeing here in the eastern United States. But upon closer investigation, we're starting to actually see some of the sexual fruiting bodies, the parathecia, embedded in these stromatic tissues. And you can kind of see these parathecia here and here, here and here. So we can start to understand it a little more because we can better describe it looking at both the sexual form, the spores, compare it with other known species, and start to really delimit those species boundaries. When we think about Ophiocordyceptaceae, again, this map may look similar to the one I showed for the Cordyceptaceae, but there are differences. Of course, there's a lot of observations in South America, in North America, in Europe, East Asia. And of course, the diversity of these fungi there are pretty profound. My colleague Joao and Charissa de Becker, and they've done a lot of work on the Ophiocordyceptaceae, and there's certainly a lot in the tropical rainforests of South America, but we can find examples as far north as Massachusetts in the eastern United States. And I just received some samples from a colleague to help identify those things. So the Ophiocordyceptaceae includes greater than 10 genera. Ophiocordyceps is clearly one you're familiar with, and really the inspiration behind The Last of Us. But things like Purpuracillium, which is an anamorphic state, it looks like a purple penicillium, and maybe hence the name Purpuracillium. Talipocladium is one that you're probably familiar with, just because that is the fungus from which cyclosporin was discovered. This also includes domesticated mutualists of Hemiptera, especially cicadas, as well as plant endophytes. A couple of years ago, John McCutcheon's group discovered that a number of cicadas that typically would have bacterial partners in their abdomens to supplement their diet because of their poor choice of food, a sap diet. They supplement amino acids by having bacterial partners. Some of those Hemiptera have supplanted their bacterial partners by domesticating Ophiocordyceps. And that was a really interesting discovery because, you know, we think of Ophiocordyceps as these parasites, but some insects have learned to tame them and use them and benefit from them. Ophiocordyceps unilateralis, this is the zombie ant fungus known and probably among the most well-known example. This is a cartoon from a recent annual review of microbiology by my colleague, Charissa de Becker. And it kind of shows the way that these zombie ants are being infected. If we start with the actual infected individual with the stroma and the sexual spores raining down onto unsuspecting ants below, those ants become infected. It affects with their normal rhythms. It hinders their ability to communicate with members of their colony. It causes staggering and disorientation, which leads to hyperactivity, summoning behavior, and then affixing death, after which the fungus is able to erupt post-mortem and continue that cycle over and over again. And this is the work of the master puppeteer, Ophiocordyceps, which coerces its host to climb to those exact positions in the right humidity at the right distance to really maximize spore dispersal. They are just puppets of their master, the Ophiocordyceps fungus. here we can see some examples of the same fungus. This is the Ophiocordyceps Capinodii floridiana, and we could see the sexual fruiting bodies, some of those sexual spores. We could see the flask-like parathecia that are embedded inside this stromatic tissue, just to get a view of how these things actually look in real life and a cartoon depicting of how it happens. Now, I don't study Ophiocordyceps per se, but recent work on looking at contemporary threats to maple stands in the eastern United States led me to an unusual discovery. And what was that? Well, we were looking to see what fungi were colonizing stressed and healthy red and sugar maple throughout the central Appalachian region. And we actually found a number of Talipocladium, which is in the Ophiocordyceptaceae, Talipocladium species forming novel lineages, indicative that they're actually unique species, unnamed species, that are basically colonizing the bark of maple trees. And this was the most abundant fungus we recovered from maple trees. And you could see one clade called Talipocladium species 2 here, one called Talipocladium species 1 here. And they were both from asymptomatic and symptomatic trees, both from red maple and from sugar maple. And what's really interesting is at the same time we were uncovering this, there's been some reports of declines in a native longhorn beetle called the maple borer. And it really raises some questions of whether or not these Talipocladium that appear to be endophytic in some of these native maple stands are making the stands more resilient to attacks by insects by forming protection or actually having a facultative entomopathogenic lifestyle. There's more work that needs to be done, but it is a really interesting first step, and it does raise questions about the dual lifestyles of entomopathogens and how they're serving these roles, sometimes preemptively, and providing resilience to their hosts. If we look at the Claviciptaceae, we can see the map spread out here. Less dense distribution across the US and South America, definitely lower incidence. In Southeast Asia, lower incidence. Does that mean they're not there? No, it just means that, you know, contemporary observations are limited and maybe highlighting needs to study these things more. Now, the Claviciptaceae is quite diverse. It includes some 50 genera, including Metarhizium, Claviceps, Conoidea, Pechonia, Epicloe, Balantia, Paraglandula. You might have heard of Epicloe and Paraglandula because these are well-studied endophytes of grasses and morning glories, a flowering plant that we have here and probably there as well. Of course, I'm sure a lot of you are familiar with Claviceps purpureae, one of the most famous plant pathogens, but also, you know, the causal agent of ergotism, which is an important disease of cattle these days, but in you know, in developing nations where food testing is not as rigorous, these sclerotia can make their way in and provide, you know, produce mycotoxins that could you know, affect us as humans. Metarhizium is a widely used biocontrol and used in various parts of the world under different names and in different formulations. Now, I study, or I have in recent years studied the fungus Conoidea ludio rostrata. And this is in the Claviciptaceae, obviously. We could see it here sporulating atop elongate hemlock scale crawlers on the underside of true fir needles. And we kind of discovered an epizootic of this fungus on elongate hemlock scale in Christmas tree plantations in North Carolina in 2020. And this really raised some questions of whether or not it'd be useful as a biocontrol. The fungus actually was first reported on whiteflies in Louisiana in the early 20th century, but we have yet to establish DNA sequence links between these early herbarium specimens and our contemporary samples, and we're currently working on that. Of course, with a lot of things, we want to sequence DNA because we want to build family trees and ask and answer, how are you related to what we know of this fungus? And here's our Conoidea crella grouping with other Conoidea ludiostratas. And here's our isolate grouping with some Japanese isolates from whitefly, which, you know, will raise a question of whether or not our isolate is actually originated in Japan on whitefly and made its way to the United States. I think with, just one locus here, more sequence data is needed, microsatellite, whole genome sequencing to make those kinds of conclusions. But it allows us to come up with some testable hypotheses, which are, useful. One of the other things we could do in the modern day is we could sequence genomes, and that really allows us to answer what potential do you have to produce interesting secondary metabolites. I started off this talk by telling you that the entomopathogenic fungi produce some really remarkable secondary metabolites. And that is no different for Conoidicrella luterostrata. If we look at our fungus here on the bottom row, this bar graph denotes the number of biosynthetic gene clusters capable of producing interesting metabolites. And you could see, compared to even something like Metarhizium, which is known to produce a lot of secondary metabolites, or Claviceps purpureae, which is known to produce these ergot alkaloids, Conoidicrella produces far more than any other species included on this list. In fact, we found 48 biosynthetic gene clusters. And that includes gene clusters, intact gene clusters for something called cyclopiazonic acid, which is a neurotoxin, as well as cephalosporin C. You might not know cyclopiazonic acid, but if you've ever had a bacterial infection, you might have taken a cephalosporin-like antibiotic, which is really useful towards bacteria. And finding cephalosporin C in another fungus unrelated to the ones previously found in is useful and really interesting and probably worth pursuing some more. We are now currently pursuing some Ashersonia and Balantia work, looking at links between historic herbarium specimens and Conoidicrella, but also looking at some of these interesting members of the Claviciptaceae that do some strange things to their grass hosts. Well, this is called black choke disease. And you can see this beautiful stromatic tissue just starting to kind of girdle those stems. Now, the Zoopagomycota is really where my specialty lies, and this includes the Entomophthalales. We could see the Entomophthalales here form a close allyship with the Kixellomycotina and the Zoopagomycotina, forming the phylum Zoopagomycota. Now, these are among the earliest diverging non-flagellate fungi. If you go back further in evolutionary time and to the earliest diverging lineages of kingdom fungi, you find a lot of flagellated or swimming fungi like the chytrids. Zoopagomycota and the Entomophthalales specifically includes 15 genera. Arthrophaga, which is a millipede specialist, Entomopthora, Furia ithacensis, which is specific to snipe flies, Mesospora, which I study on periodic and annual cicadas, Pandora, Strongwellsia, Zoopthora. These are all really interesting genera, well, especially to me, but include the zombie fly fungus, Entomopthora musci, and Mesospora cicadina, which are among the most well-known. The distribution and the incidence of reports of these are less, but still some good observations on several continents, highlighting where we need to look more and where they may not occur. Of course, I think a lot of people that know my work know that I study Mesospora, the zombie cicada fungus. This was established by Peck in 1879 and includes some 12 obligate sexually transmissible species. Here's a phylogeny we published in 2020 showing the relationships among the known sequenced Mesospora species. A really interesting aspect is we determined that two formally described species, Mesospora levospora, seen here, and Mesospora platypedia, seen here, are actually one species that infect two different genera of cicadas. And recent work that I've been doing over the summer actually shows that the Mesospora that infects periodical cicadas is actually two distinct species that follows the evolutionary history of its host. And that's work that's currently underway in my lab. Of course, what gets people excited about Mesospora is the fact that, we found something that could explain the modification of behavior, and that is that Mesospora cicadina in the periodical cicadas produces cathinone, which is an amphetamine, a naturally occurring one that's found in cat plant. And it's also found in Mesospora cicadina plugs. And we also found the psychoactive tryptamine, psilocybin, and Mesospora levospora plugs. So really, we're able to use analytical chemistry to confirm these things, but the genetic underpinnings of these are very difficult because we're dealing with very big genomes. But the zombie cicada fungus has yielded really interesting secrets that we didn't know before. One is that we found the first detection of amphetamine production in kingdom fungi. We found the largest fungal genome. If you look over here, you can see the breakdown of genomes by order or by phylum, and only rusts really compared to those in the Entomophthalales. And the large genome is 1.5 gigabases. Despite its large size, it has so few genes, which is really an interesting yet unconfirmed. We really need transcriptomics to confirm these things. And it's full of these transposable elements. So it's got all these kind of repeat sequences that are often considered non-self, and it raises the question, what effect are they having on the evolution of these obligate biotrophic parasites of insects? So that's some of the work I've done on that. I've also done a little work with Carolyn Elia on the zombie fly fungus. And this is trying to, and something I've done and continue to do, is link those historic morphological characteristics with the current DNA-based approaches. And really what that work shows is that, at least in the Entomophthalales, you can't really rely on these morphological characters because there's so much overlap and there's so much confusion when you kind of juxtapose that with the molecular data. So, it's really important that we generate molecular data for these different species to see if those species boundaries actually hold up. I'm currently working on Arthrophaga, which right now is currently a monotypic species, but data suggests that there's at least 3 species in the United States of this. And I'm working on Pandora, which is an Entomoptera-like summit disease pathogen of blowflies at higher elevations in West Virginia. Not too much data on that, but still an interesting group of fungi that I hope to continue to study. But when I wrap all this up and think about what we've learned from entomopathogenic fungi, it's that there is a solid foundation of work on entomopathogenic fungi, but only for known species in both these orders, the Hypocheiales and the Entomophthalales. But huge gaps still exist. Majority of entomopathogenic fungi have yet to be discovered or described. That's true of all fungi, but it's obviously true of entomopathogenic fungi. The production of diverse secondary metabolites like cathinone and cyclosporin, really suggests that these interactions may be the last or the next frontier for drug discovery and have real utility in human medicine and pharmaceuticals. And I think what this also shows, based on the maps I was kind of showing you, is that iNaturalist users, that is naturalists without any scientific background, just a curiosity to observe, are really helping to accelerate the work that we do as mycologists, because they're providing contemporary locations for specimens that we can then go sample and work with them to teach them more, but also learn from them and about the habitats that they study and spend time in. But with that, I think that wraps up my time. I only have a few minutes left, and I'm going to stop sharing. And thank you for your attention today.
Thanks, Matt. That was super. Always exciting to know about these fungi. We look forward to questioning you later on.
Thanks, Lynn.
Okay, so next we've got Bob Blanchette, who's a professor in the plant pathology department at the University of Minnesota. As I mentioned at the opening of the session, Bob has actually spoken in a previous incarnation of our global series that we have each year. Bob's interested in all sorts of things: temperate forests, tropical forests, the Arctic, Antarctica, so a true global player. And over the years, he's studied many different areas of mycology, including wood decay fungi. He's got current projects in biocontrol using fungi and many studies on ethnomycology. And today he's going to tell us about historic uses of forest fungi. So over to you, Bob. Welcome.
Thank you very much. And very nice to be with you today. And I'll get this, there we go. Today I'd like to talk about how fungi were used in the past, and they have been used for long periods of time. And if you listen to the session 2 today, Professor Abdel Azim had talked about the fungi used in ancient Egypt. And in that case, this was used 2,000 to 3,000 years ago in the depiction of fungi, likely psilocybin or psilocybe, in these mushroom carvings in the stone in various temples. The fungi that I'm going to be talking about today are all polypores. They're fungi found in forests, and many of these polypores can be perennial, like you see here, which will grow year after year, and some others here are annuals. They all have in common that they produce pores, and of course, their production is to produce some of these basidiospores and to produce spores that will cause new colonization to take place. And so if you take a look inside one of those pores, you'll find basidia and therefore basidiospores that are going to be shot off and bring the fungus to new areas. And my interest in ethnomycology really got started several decades ago as I was working in American Museum of Natural History on a wood decay project. And this involved looking at Western red cedar, and many of these old objects from the 1800s were severely decayed, and looking at some of the interesting fungi that were causing the decay in these. And at the time, an object came up that I thought was very curious. And this carved object was used by shaman. And the object here was at first thought to be wood. And you can see it has annual layers in it. But this particular object was not wood. In fact, it was a fungus commonly called agaricon. And this is Larissiformes, in the old name Fomitopsis officinalis, that occurs in the western United States and the Pacific Northwest. It can produce really large perennial fruiting bodies. And in this case, this one being almost 100 years old, year after year, putting new layers on as it grows. And when I first saw this, I said, isn't that an interesting thing that we have a fungus that has medicinal properties, and it was used by shaman. And when I told the museum curators about it, they were quite interested and started looking through the collections. And the next time I visited, they pulled out a cart and they had 6 of these in their collection, all of these carved from agaricon. Now, this shows that this fungus was used over a wide area for a long period of time. All of these in their collection were collected in the 1800s. And many groups used these, all used by shaman. And we found over the years, as we started to search various collections, that there's actually 14 of these in 6 different museums. And all of these used by various shaman to cure the sick in various rituals. And interestingly, they selected a fungus that has medicinal properties. And we know today that there are even new antiviral and new antibacterial compounds being found in this particular fungus. Also, this fungus has some interesting features in that as it grows in the wood and it decays the wood, it produces these mycelial mats within cracks and crevices. And these mats can be quite big, and they can be pulled out from the decaying wood and are often used, or were often used, by indigenous people of the Pacific Northwest. And in our search of some museum objects, we came across these, and these were wall pockets, twinglet wall pockets, and they were used to put into dwellings. And there's a pocket here that they would put valuables into. And usually these are made out of hides, deer hides, or other types of hide. And in this case, they looked a little bit different in getting a sample of these and looking at it. It certainly was not a hide, but it turned out to be mycelium, and it turned out to be mycelium mats produced by agarikon. And today we have a lot of interest in this and a lot of industrial potential uses of this, as we have these mycelial mats being made into mycotextiles and an alternative to leather. And these can be treated and sort of made into look just like leather products. And also, these mycelium mats have lots of other uses too, for packing materials to replace styrofoam and things like that. But we have this mycelium mat used by Native Americans over 100 years ago for a mycotextile. And I am sure there's more of these in various museums, and they just need to be found. Also, in the Canadian Museum of History in Ottawa, there's this object, and this is a carved mask from agarikon. And what is interesting about this mask is that it was used for various rituals during an eclipse of the sun or the moon, showing the importance of this particular fungus for use in ceremonies when a very strange event takes place. And this is from the Bella Coola village in British Columbia, and again, dating back into the late 1800s. So it shows the use of these fungi for various purposes, and in this case, having supernatural powers. Now, there are a lot of other uses, and I have been trying to find additional uses that were used by indigenous people. And this is an example by the Anishinaabe, which is a Midwestern group. And in this case, they used wood attacked by armillaria that was glowing, and they would use this to put onto their masks for their night dances and ceremonies. And also, there's a lot of legends that have been written about this. And this is all by this Anishinaabe herbalist that reported these, and legends where, in this case, a group was surrounded by some enemy forces, and someone laid out a path with the glowing embers of armillaria wood, so to guide all the people out to safety, so they could follow this glowing path away from the danger. So armillaria is an interesting fungus. It does not glow in the dark. The fruiting bodies don't glow in the dark. They're not bioluminescent. This is a photograph taken by Taylor Lockwood of Mycena. And in this case, these fruiting bodies do glow. But for armillaria, it's not the fruiting bodies that glow, but the white mycelial fan that is produced as this attacks trees. Now, in session 2, Deborah talked about armillaria, and it is a fungus that attacks trees. It will attack the roots, grow up to the base of the tree, and girdle the stem. And if you pull off the bark, you can see this white mycelial fan. And the white mycelial fan is what glows in the dark. And sometimes you can find this glowing very brilliantly as you will pull off the bark and see the mycelium glowing underneath. And also, we can grow this in culture. And years ago, a member of the indigenous community came by and was asking about how can they get material to glow? How can we find armillaria and grow it in culture and use it in various ceremonies? And at the time, I wasn't able to produce this, but now we can certainly grow this in the laboratory and get it to glow magnificently in the lab. And recently, Taylor Lockwood has put together a time lapse. This is sort of an overview as his camera is moving over the glowing mycelium, showing sort of a very eerie and ghostly look of this fungus that actually produces this bioluminescence in the dark. We also have in other parts of the world masks that are also used for various purposes, used by shaman and for used to cure the sick and in various rituals. And these come from the Himalayas. Here you can see the fruiting body of one of these large perennial polypores. And in this case, one was used, it was sort of turned upside down. The pore layer is up here, made into these masks. And in Nepal, in Bhutan, and some other places in the Himalayas, masks are used often in various ceremonies and rituals. Usually the old masks are made out of wood, but we also have found that there are some of these masks that are made out of fungi. And here again is an example where they used fungi with medicinal properties to carve these masks out of, and then they would use these masks in various rituals to cure people of sickness, to ward off illness and that type of thing. And they usually have a very dark patina on them. And usually during the, after the ceremonies or the rituals, they would place them in the rafters of the huts. And in these huts, they had open burning and cooking. And so there was smoke that would move up and turn these a very dark color. And these would be placed up into the rafters for protection. Some very interesting looking ones here. Fungi that were used for pigments. In the case of the Pacific Northwest, we have Echinodontium, commonly called the Indian paint fungus. It has a brick red color, a very distinctive coloration to it, used as a red pigment, but also in collection notes, we find that it was also used a little differently, and that this fungus was burned. And when you burn this, it turns into a charcoal that is a black powder. And they would take that black powder, mix it with some spruce resin or gum, or fat from goats or deers, and they would apply it to their faces. This is to protect from sunburn or to prevent insects from biting them, also to stop snow blindness, and was used as a sign of mourning as well. And there's a lot of examples of these in various natural history museums, some dating back to 1875, but this is a widely used fungus for pigments and also for burning and turning black and using it as a face paint. We also have the use of green stained wood. And this is more of a modern image here from the Pacific Northwest, where a green paint was used. But over 100 years ago, they also used a green pigment. And this pigment was used for various dishes and things like that. And this came from a fungus that produces this green stain in wood. And apparently, as the collection notes indicates, that the stained wood was ground to a powder, mixed with fish eggs, and used as a pigment. This is a fungus called Chlorosiboria. And this is a fungus that's in the Ascomycota. And in addition to its brilliant greenish blue apothecia here, the fruiting bodies, it also produces this characteristic color in the wood. And we find that this is a fungus that has been used in the past a lot. And this dates back to the early 1500s, where a choir stall was made. And these are intarsia panels. The intarsia panels are where wood is taken and different colorations of the wood put together to sort of give a three-dimensional image. And here you can see the image here as if you were looking out a window, in this case, looking at a cupboard with the open doors of the cupboard. And of course, this is just a flat surface, and they give it this image by using the different colors of it. And we have the use of green stain here for the landscape in these very old intarsia panels. Also, here's a cabinet from the 1700s, very beautifully inlaid with various woods, giving these images, and also the Chlorosiboria used. And in the UK, this has been used in Tunbridge ware. This is another example where I think starting in the 1800s, that boxes and other types of materials were inlaid with mosaics of different colors to give images and beautiful mosaic features on these various types of boxes or other objects. And I think to this day, some of these are still being made in the UK. In the Smithsonian, they have some interesting fungus boxes. And when I saw these listed, I was very curious to find out, well, what kind of fungus are they carrying around in all these boxes? These, again, are over 100 years old. They are from various tribes, but all from Alaska. And they are made out of wood, bone, and other materials, all to carry a fungus. And one of these in the Smithsonian had some collection notes, so we know what they were used for. And they were used to actually carry around some chewing tobacco that is mixed with the ashes of this fungus. And this is Phylinus igniarius. And when they burn this fungus, it turns into a very white powder. And they mix this ash in with tobacco, and then it is chewed. And apparently the change in pH causes the nicotine to go into the mouth and into the bloodstream very rapidly. So supposedly it gives a very powerful kick. And this is the fungus here, Phylinus igniarius, that is used, and apparently only this fungus used for this purpose. And even today, if you go to some of the indigenous communities in rural Alaska, you can find in some of the supermarkets, they have baggies of the burned Phylinus igniarius that they will use and mix with tobacco. And also there's a lot of efforts going on with trying to inform people of the dangers of this. Can you imagine, you get nicotine from smoking and chewing regular tobacco, and you put this into it, and you get this huge surge of nicotine into the bloodstream, which for health purposes is not good. Also, as we searched through various natural history museums, we came across some other objects that appeared to be fungal. In this case, we have these beads, necklaces of these beads that were thought to be carved aspen roots. But in looking at these, they didn't seem to be wood. They appeared to be fungal. And taking some samples of these, we certainly found out that this was not wood. These were fungal beads. And looking closely at them, we found some basidiospores present in them. These were carved fruiting bodies of a fungus called haplopores. And the basidiospores are very distinctive in their ornamentation of this particular fungus. And this is what that fungus is. It is a fungus that occurs in the northern plains, a rare fungus now, and it also is a fungus that has a magnificent aroma. And as we searched through various museums for these, we found that there are some photographs in various collections that show how these were used. Here we have 3 chiefs of the northern plains that have their ceremonial robes. And on the ceremonial robes, you find the haplopores beads. And again, these have a very strong aroma to them. In fact, I have one here in my office that I've had for several years, and this still has the aroma. And this fungus produces anise aldehyde, so it has a very, very strong anise aroma. But there's lots of evidence of these in various images from old collections as these are used. There are also examples where they are in medicine bundles and the fungus was used medicinally. And we know that it also, in some places, is still used today. And I had the opportunity to visit a Woods Cree healer in northern Alberta. An anthropologist, David Young, was working with the Northern Cree and saw some of the publications that we had written about the fungus and said that the group that he's working with appears to also use a fungus that's very similar. So we went to visit Russell Willier, and the fungus that he uses and the information that was passed down from his grandfather is the haplopores. And he uses it as a smudge. And as he says, the fungus opens the door to the spirit world and allows me to see and hear the spirits. So this currently, in some places and also in the past, was used to ward off sickness and to help people prevent sickness as well. In the Scandinavian countries, the Sami people, haploporous also occurs in some of those regions, and they used it a little bit differently. And there's some collection notes that Carl Linnaeus made back in 1737 as he made expeditions up into the northern Arctic region. And he indicates their use there being different. The Sami men engaged in courting use the scented fungus haploporous, and they store it in a pouch and wear it on them in order to be more pleasing to the women due to the sweet fragrance that it sends forth. And it is a remarkable fungus, and it holds that aroma for long periods of time. As I mentioned, I have one here, several years old, and it still has a very, very strong aroma. Now, another fungus that has been used a lot in the past is Ganoderma. And Ganoderma lucidum is actually a name for a complex of these laccate or shiny Ganodermas. Here in the United States, we have 12 different species of fungi that will fall into this group. But in Asia, it's often called reishi or lychee, and commonly called the fungus of immortality. And we know that this has medicinal properties. It has been used in Asia for long periods of time. Today, we find this in tea. You can have reishi tea. You can find reishi lychee in coffee. Many different sources of this, drinks, and even in chocolate. So lots of uses for this. In the past, there have also been a lot of uses for this in China, and I had the opportunity to work at the Forbidden City with the Palace Museum and the World Monument Fund. And there we were looking at some very old buildings. These are wooden buildings that were built by the Qinlun Emperor, who reigned from 1735 to 1796. And he made 27 pavilions and structures in the Forbidden City as sort of his paradise palace. And there, as you can imagine, these old buildings that were actually closed for long periods of time after the emperor has died, that these were closed and not open, and now they are conserving them and now reopening them. And we were there to look at some of the decay fungi that were present, lots of brown rot, as you would expect, in buildings that are over 250 years old. But also working there, I found some interesting information about Ganodermas. And one thing I thought was amazing is that the Qinlun Emperor used to love to go on forays, and he would love to go on expeditions. And he would go collecting the lychee or Ganoderma mushrooms. And here is an old drawing, painting of the emperor made in 1734 as he was going out collecting. And we do know from looking at old paintings that we can find this was one of the emperor in the 16th century holding a Ganoderma. We also have many other types of paintings where they're using Ganoderma. They're holding it here. Here is one with a big long stipe. And this one, these two date back to 894. So the Ganodermas we know were the fungus of immortality. They were used for medicinal purposes, but they also serve as symbols. And they serve as symbols for good longevity and good health and prosperity, and also to bring good luck. And the Qinlun Emperor would surround himself in his palace with lots and lots of things with images of Ganoderma. If this brings good luck, he had lots of it around. And here is a vase here with Ganodermas, a table, a carved table. And when you look closely at this, the entire table is full of these carved Ganodermas. Paintings, most of the paintings would also have images of the Ganoderma present in them. And also they have a lot of these ruis. And these are carved out of wood and bamboo in images of the Ganodermas. And again, they're used as symbols of long life and good luck. And the Palace Museum has hundreds of these that were presented to the emperor over time as gifts. And apparently this gift of wishing him good luck, long life, and good health. But what I thought was really remarkable is that there are these very, very large Ganodermas that the emperor made into cabinets for them. And here you can see one very large one. I assume these are Ganodermas that the emperor collected, and in this case, put into a very beautiful rosewood cabinet. Here's another one. So he really liked Ganodermas and used many of these in his palace. Here's another that's currently being conserved because it has slipped from its mount. And there are 4 of these in the palace. And this one is interesting because it has some notes with it that this was presented to the emperor from his general. Can you imagine giving the general giving the emperor a gift, and the gift is this big, big Ganoderma in a very, very beautifully carved cabinet here to hold it. Also, the emperor wrote poems on the bottom of these Ganodermas, and he wrote them in gold ink. And this is just part of one of those poems. And I just wanted to put this here just to show you what the emperor thought about these fungi. And he actually thought that they were as rare and as valuable as purple sandalwood or white jade. So the display of these, I think, is really a good idea. Here's another one here, a beautiful Ganoderma being displayed on a wooden mount, and the wooden mount is actually carved Ganodermas. And I like this idea a lot. I have also mounted lots of these in my office. You probably can see in back of me, I have several of these sitting there, all bringing me good luck, long life, and good health. And I have one other one that I want to mention here, one other example of a fungus in a natural history museum. And this focuses on this area here in East Papua. And back 100 years ago, this was Dutch New Guinea. And this was sort of like an unknown territory. And lots of expeditions were being made to go out to the site to study the culture of the people there. And during one of these, by the Peabody Museum in Harvard, they found several objects and were brought back and put into the collection there. And this is one of those. This is really one of the most unusual fungal objects I have ever found in one of these collections. And this was collected back in 1925 by an anthropologist, Patrick Putnam. Again, he had gone to this area to search and look for objects of interest in their culture, and brought it back to the Harvard Museum. And there it sat in storage for many, many years. Now, this is a Ganoderma that is very attractive in that it's red. It was used as a necklace. It likely came from the neck of a shaman or the chief. But what's so unusual about this Ganoderma is it has secondary growth, and the secondary growth looks like human fingers. And when you look at a regular Ganoderma, this is what normally occurs, and that's the fruiting body, and then it stops. In this case, you had this fruiting body produce another layer of growth, and these projections all looking like fingers with fingernails. Very unusual looking. You know, you can imagine the sort of supernatural effect that this would have when a shaman would be wearing this. Now, we do know that this fungus, you can grow it in culture and you can get the regular sized fruiting bodies here. And we also know that these finger-like projections occur when it is trying to get up out of the ground or out of the leaf litter. And so if you have high CO2, then it will produce these projections. And I've been trying to mimic the production of these fingers on one of these, which is not easy to do, but in some cases it does occur. In this case, we had some success where we have the fruiting bodies, and then you enclose it, you increase that CO2, and you get those fungal-like fruiting projections coming out of it. Now, this is really a wonder of the fungal world because we have all of these in one plane. And I think what's happened here was that when this was on the tree fruiting, the tree fell over and somehow it got covered over. Maybe there was a flood and there was a lot of sediments, or there was a lot of organic material, and the fungus produced new growth to try to get up out of it. But then that new growth had all of these very interesting features of looking like human fingers. So lots of examples of these various fungi in various natural history museums. As these collections get digitized, I'm sure that more and more of these are going to be found. And I want to acknowledge all the help of curators and conservators at these museums to help find the ones that I have just been talking about. And if anyone is interested in any publications of this, you can find them at my website. And I have those examples there, as well as others that I didn't get a chance to talk about. And with that, I'll end this, and we'll move to the next person.
Thanks for sharing that with us, Bob. Absolutely amazing. Really, really super. Thank you so much. And I'm sure that I hope there'll be questions later. Next speaker then is Patricia Velez. And Patricia is at the National Autonomous University of Mexico, where she's a research professor and she teaches there. She's interested in diversity, ecology, especially in marine ecosystems. And of course, relatively few people study fungi in marine ecosystems. So it really is a great joy to have you with us. So Patricia is going to tell us about diversity, ecology, patterns and utilization of deep sea fungi in Mexico. Over to you, Patricia. Welcome.
Thank you so much. I'll begin sharing my screen. Okay. So, well, thank you very much for the kind introduction. And thank you also to the British Mycological Society and the organizers for the invitation. And well, today I'm delighted to share with you some of our research and recent work on the diversity and ecological patterns of deep sea fungi in Mexico. So the presentation is organized as follows. We'll begin with a brief introduction into the oceans, the definition of marine fungi, some examples of deep sea ecosystems in Mexico, and let's move into fungal diversity in the deep sea, using as an example 2 different ecosystems, deep sea vents and oxygen minimum zones. So using these 2 examples, we're going to discuss some of the fungal functional roles, metabolism of hydrocarbons, and antibacterial activity. So, well, the oceans are the lifeblood of planet Earth and mankind. They're actually very good at sequestrating carbon dioxide and also regulating the temperature of the overall Earth. They also flow over nearly 3/4 of our planet and hold most of the water on it. So they also sustain the productivity of a number of countries, especially those that are coastal or island nations. And so the sustainable utilization of oceans and their conservation is very important for global economic growth and food security. However, a combination of factors are presently threatening a new mass extinction event on the oceans. Here in this slide, I listed a few examples, including the acidification of the oceans, the overexploitation of the resources, of course, pollution that usually leads to hypoxia, and climate change. So the joint effect and the cumulative impacts of these factors on the ocean seems to be greater than estimated. However, still it is poorly understood. So under this scenario, obtaining baseline data on underappreciated species is very important. So we need to understand the actual magnitude of marine fungal diversity at both taxonomic but also at the genetic level. We need to understand the distribution of these organisms, and also we need to generate a lot of information on their response to stressors. So marine fungi are widely distributed across all marine ecosystems, including sandy beaches, coral reefs, mangrove forests, the water column, and of course the deep sea. And today's talk is going to focus in this deep sea. Marine fungi are an ecological group instead of a taxonomical group. This ecological group includes a taxonomically diverse group of microorganisms that are able to adapt, evolve, grow, and sporulate under marine conditions. They include mostly saprobiotic species that play a very important role in nutrient cycling. However, also pathogens have been described. And despite the large area that marine ecosystems cover in our planet, currently we only know less than 8% of the described fungal taxa coming from marine systems. And these organisms inhabit very fragile ecosystems that are susceptible to anthropogenic stressors that lead to changes in several key environmental variables, for example, pH, temperature, nutrients, carbon dioxide, and of course, salinity. So these stressors have been preliminary based on few works demonstrated to cause reductions in diversity that may be leading to changes, functional changes in the ecosystem. However, this of course remains poorly understood. So we need more mycologists that work in marine systems. And particularly going more deep into the deep sea fungal diversity, since the first isolation of fungus from the deep sea more than 50 years ago from the Atlantic Ocean, the investigation of these organisms has been increasing, but not so much. Most of the species remain to be described, to be investigated, but what we do know so far is that deep sea fungal diversity resembles a lot terrestrial taxa in terms of the taxonomic identity of the organisms. And these microbes rely on the precipitation of organic matter from the upper water column. This organic matter is known as marine snow. I included here a couple of images in case you're not familiar with how marine snow looks like. So just to remember, deep sea ecosystems are extreme for life. They may be considered as those ecosystems that occur at depths higher than 1,500 meters below the sea surface. They cover more than half of the Earth's surface. They are generally cold, except for hydrothermal vents. They are always under absolute dark, and hence they are oligotrophic because primary production cannot rely on photosynthesis because of the lack of light. They are also characterized by high hydrostatic pressures, and they are very important in global carbon cycling and represent an alternative to mining on land. And these unique ecosystems harbor an unseen biodiversity that might pose benefits to human health and industrial activities. So fungi that live under these very extreme conditions must cope to support them. So moving on into the two examples that I'm going to share with you today, we have on the one hand hydrothermal vents that are that actually represent top productive ecosystems on Earth, and they host an exceptional microbial diversity characterized by chemio-lido-autotrophs bacteria, but also by heterotrophic microorganisms as fungi. On the other hand, we have oxygen minimum zones that are characterized by a very low saturation level of oxygen. And also, despite this condition, they are very rich in microbial communities with ongoing anaerobic processes that impact the productivity and climate balance of the ocean. These two systems are not supported by photosynthesis. Therefore, microbial communities in these two systems sustain life and, of course, drive ecosystem processes. However, most of the investigations in microbiology in these two environments have focused in bacteria, neglecting fungi. So, well, yes, of course, we have endemic fungal species from the deep sea. Here I included a couple of examples, specifically this in the middle, were very elegant investigation describing an endemic deep sea fungus. You can see here the images. However, most of the investigations are conducted using these culture-independent approaches with massive sequencing, and these results have revealed the high abundance of potentially novel phylotypes, uncultured phylotypes, and we believe that they may be playing key functional roles in the deep sea. However, this is unknown still. So, well, why should we care? So, well, the potential applications of deep sea fungi are very wide. For example, in terms of the discovery and isolation of natural products with antibacterial and anti-cancer properties, and also because these fungi may have the ability to buffer the impacts of humans on the earth. They have the ability to use and metabolize recalcitrant polymers, therefore they may be useful for the bioremediation, and some early evidence suggests that they even may have the ability to degrade plastics. So we can only hope that if humans are unable to solve environmental crises on our own, well, fungi might eventually evolve to do it for us. So this is a map of Mexico. Here's the content, America, and as you can see, we have several deep sea and marine ecosystems. Today's talk is going to focus on these hydrothermal vents, these two oxygen minimum zones in purple, and some evidence coming from the Gulf of Mexico in oil drilling areas. So the samples were collected on board of a research vessel in collaboration with MBARI, with the help of a ROV, the remotely operated vehicle that has these robotic arms. And it collected sediment cores. On board, we collected subcores from these cores and transported them to the laboratory. We used a complementary approach using both classic microbiology but also culture-independent approaches to analyze diversity. So why did we obtain the fungal isolates? Well, because we can approach the ecology of the organisms, we can test their abilities for their potential utilization in bioremediation. We can also screen their potential for antimicrobial bioprospecting and conduct a bioactivity-directed chemical study. So here's a more close-up on the map. You can see the two different vent systems. On the one hand, we had white smokers that are characterized by calcite. You can see here the color white. Very importantly, they produce aromatic hydrocarbons. Please keep this in mind. And on the other hand, we have black smokers. You can see here the black smoke coming out of the vent. And well, also it's important here to mention that animal communities are very different. Bacterial communities are also very different, and the chemical and biological structure of these two vent systems are clearly different. So what we observed based on the ITS metabar coding of the sediment samples coming from these two vent systems and the oxygen minimum zone is that fungal diversity is somehow high. And very interestingly, you're not meant to read all these small letters, but to appreciate the huge diversity. And these ASVs that you may see, they occur widely across all sites, all three different systems, and in a very dominant fashion. They are the unculturable deep sea phylotypes. So this agrees with several works from deep sea reporting the presence of these uncultured phylotypes. So when we investigated the oxygen minimum zone in the coast of Mazatlan in the Pacific Ocean, we observed the same pattern. These deep sea, extreme deep sea systems host several uncultured fungal phylotypes. This work was done by a master's student, Judith Posadas. So next we moved into the culturomics of these sites. So we obtained 32 axenic fungal isolates clustering into 19 OTUs, including all of these genera, and very interestingly, we observed as a top abundant species Aspergillus idoides. You may remember it for its pathogenic relationship with corals. So we used these isolates to investigate further into their functional roles in the deep sea using stable isotopes of carbon, nitrogen, and sulfur. So what these results indicated is that we have two different ecological groups occurring in the samples. The first one relying on mixed carbon sources. This means that the carbon that was originated in the upper water column result of photosynthesis that precipitates into deep sea. And the second group relies exclusively on carbon that is fixed in the deep sea. So this is very interesting. And these organisms are acting at different trophic levels. And in terms of nitrogen, they are also relying on both local and external sources. So we also identified the assimilation of bacterial and animal necromass acting as parasites. So please also keep these cross-kingdom interactions in mind, because we went further into detail with the research, the master's student, Jessica Hernandez, that investigated using comparative transcriptomics, the cross-kingdom interactions of fungi and bacteria from deep sea vents. And in summary, you can see here in this cartoon that based on her results, it seems that bacteria are attaching to fungal hyphae and breaking down fungal wall, which results in the leaking of fungal content, so bacteria are feeding on hyphae. As a result, the fungus synthesizes cell wall and membrane elements to fix that, and also attacks the bacteria using producing several antibiotics. So we conclude that this fierce antagonism between both fungi and bacteria might be categorized as mycophagy. And so we wondered what if beyond nutritional relationship, this interaction could represent an advantage for any of the two organisms. So this is part of undergrad investigation of Blanca Martinez, who designed a acrylic maize. So we inoculi used this maize and grew fungi in monoculture and bacteria, and also the co-culture. and we observed a hitch-hiding pattern where bacteria attach to the hyphae, fungi resolve the maze very quickly, and at the end of the maze, bacteria jump off the fungus. So it seems that bacteria are taking advantage not only of the filamentous growth of the fungus, but also because of they can feed on the fungus. So it's very interesting, this cross-kingdom interaction. And well, lastly, moving into the last part of my talk, let's go into the potential utilization of these genetic resources. So early evidence coming from the Gulf of Mexico, where sediment samples from oil drilling stations were collected, we isolated fungi and tested their tolerance towards two different hydrocarbons, an hexadecane and hexadecene. And we observed in the use experiments that some of these species are able to grow, tolerate, and actually grow better in relation to the control when the culture medium is supplemented with the hydrocarbons. So we looked at this data using transcriptomic differential gene expression analysis and identified two groups of genes. Those that were downregulated when the fungus is using glucose as carbon source, versus those genes that are upregulated when the fungus is using the hydrocarbons as a carbon source. So we were able to confirm the utilization of these two hydrocarbons as unique carbon sources and the potential use of these fungi in the bioremediation of oil. So next, this is work done by the postdoc Lucia Romero. We tested the metabolism of complex hydrocarbons. In other words, crude oil. So we tested light, medium, heavy, and extra heavy crude oil. And you can see here in the SEM micrographs, the fungal hyphae penetrating the matrix of the oil. In this experiment, you can see this is a drop of oil and the fungal mycelia is surrounding the oil. And these are images of the sensitivity assays, where you can clearly see that fungi are not so sensitive to the presence of these toxic elements. So we run an experiment using high pressure reactor coupled to SARA and HPLC. And we were delighted to observe that these results are for extra heavy oil, crude oil. You can see here, this is where we inoculate the oil, mineral medium, and fungal conidia. And you can see here in the x-axis, the composition of the oil. And if we move to the left, to the right, you can see the heaviest portions of the crude, of the oil. And as a result of fungal action, these are used. So this is unique because usually bacteria cannot attack these portions of the crude oil. So it's very promising. And well, lastly, these are the results for the antimicrobial bioactivity of the isolates. And you can see here that most of the isolates showed antimicrobial activity towards this escape or typically used pathogenic, human pathogen bacteria. And what is very interesting is, well, first of all, this agrees with former culture independent studies that hinted at notorious antagonic roles. These are based on fungal analysis and also the isotopic analysis. So we can conclude that these fungi are also prominent antimicrobial producers, and they are able to target several pathogens. So they are also confirming the important role of these molecules in cross-kingdom interactions. As I already showed you the results. So in terms of the directed biochemical study, we were able to identify these several compounds with antibacterial, antiparasitic, and anti-cytotoxic against leukemia, breast cancer, colon cancer, hepatocellular, and cancer and malignant melanoma cells. So it's very promising, these results, at notorious bioprospecting potential. So further works or further investigations should implement the OSMAC, one strain, many compounds approach, to trigger the expression of silent gene clusters. And some work, research groups in China have demonstrated that using heavy metal stress is very good for this, in this sense. So in conclusion, hydrothermal vents are hypodiverse extreme systems for fungi. Fungi are key players in ocean-level biogeochemical cycles, and they are able to act at different trophic levels. These organisms are valuable poly-extrematolerant genetic resources with a potential use for oil bioremediation in the ocean. They have a strong antibacterial activity against human microbial pathogens, targeting some species, for example, Aspergillus acpi, that's able to target several pathogens. So further investigations in bioprospecting should benefit from stress-driven strategies to enhance the discovery of novel molecules. So with that, I'd like to end my talk. I'd like to also thank the following people and institutions for funding, and also all of my colleagues and students. Thank you so much.
Oh, thanks, Patricia. That was absolutely fascinating. I think fascinating is my overused word of the day, but nonetheless, it's very true. Absolutely intriguing. Thank you so much. So now we move on to our last talk, and not just of this session, but indeed of the whole webinar. And it's going to be given by Michael van Nuland. Michael is the lead data scientist at SPUN, the Society for Protection for Underground Networks, which I think he is based in Oregon. He's an ecologist, evolutionary biologist, and he combines data sources, data science, large-scale observations, experiments, all sorts of things to try and explore and understand broad ecological patterns. So, Michael, please share your screen with us, and over to you. We very much look forward to hearing about mycorrhizal biogeography.
Awesome. Thanks so much. And thanks, Lynn, and to the British Mycological Society for the invitation. It's a real pleasure to be here today with you. So I'm going to share some work that we've been doing at SPUN, mapping mycorrhizal biodiversity and using that type of spatial information to understand where these types of symbioses might be at risk on our planet. And fundamentally, this is going to be a talk about mapping, which is personally one of my absolute favorite topics to cover because maps are so fascinating, right? They can be works of art, they can reflect history and culture, they can hold key insights for science, and they can do all of this simultaneously on a flat, two-dimensional surface. So they're these incredibly powerful tools for sharing information. And so it's no surprise that mapping nature has really been fundamental to how we understand the Earth's ecology. Some of my favorite examples of this are these botanical geography maps that were inspired by an early naturalist, Alexander von Humboldt, and they contain these really highly detailed illustrations of plant species along mountainsides all across the world. Now, in many ways, these maps were the catalyst for the entire field of ecology that we know today. And they were so impactful because people saw these maps and they started asking questions like, why do plant communities change across mountain ranges? Why are some species only found in certain habitats and not other? Why do some crops thrive here and not there? Now, fast forward to the present day, and we have gotten incredibly good at mapping the above ground world, right? We have expansive plant surveys. We We track animals with GPS collars. We have camera traps and audio traps set up. We have highly detailed forest inventories taking place all over the globe. And now we're even using drones and satellites to measure changes in vegetation from space, like the image here from the latest Landsat mission. All of these efforts, from illustrating plants on a representative mountain range to capturing satellite images of the world, all of these efforts have enhanced our ability to not only describe the biodiversity of the planet, but also to understand how ecosystems function and produce CO2 or draw carbon down or change through time. The issue is that most of what we know about from the natural world comes from this overwhelmingly above ground perspective. And as a result, we know so little about the distribution and the diversity of soil organisms like soil fungi. And these are the same types of organisms that we're learning really control so much of what we see and rely on above ground, whether that's improving plant health or overall ecosystem stability or cycling nutrients, etc. We really need spatial data. We need maps of where species that live below ground are in order not only to understand how biodiversity is fundamentally arranged on this planet, but to understand how communities and ecosystems are working now and how we might help them become healthier and more resilient into the future. And at SPUN, we think mycorrhizal fungi are a really key group to focus on. And that's because they form these physical connections between fungus and plant root systems. So they're this great conceptual and direct way to link the above and below ground worlds together. So mycorrhizal fungi are a type of symbiotic fungi. They form relationships with plants and specifically plant root systems. They're incredibly widespread across plant groups. About 85% of plant species on the planet form some type of mycorrhizal association. So the vast amount of plants that you see on your walks in your local parks are likely associating with some type of mycorrhizal fungus on their root system. There are really ancient symbioses too. There's a particular type of mycorrhizal fungi that we think helped plants colonize land in early Earth's history by basically functioning as an early root system for plants. And they are defined by this reciprocal exchange of resources. So the plant is photosynthesizing, building these carbon structures, carbon sugars, and they're sharing some of that carbon with their fungal partner below ground, who is building these hyphal networks through the soil, gathering and mining resources and essential nutrients from decomposing organic matter like leaf litter, and they're trading those resources to the plant in exchange for some of that carbon. And through these vast hyphal networks that they make through the soil, they can contribute a significant portion of the living biomass of soil environments. Now, an important function of these immense hyphal networks is the distribution of plant carbon below ground. So plants are absorbing CO2 through photosynthesis, but they don't use all of that carbon to grow and reproduce themselves. They're trading a significant portion of it to their fungal partners, like I was mentioning. And so that means that mycorrhizal fungi are really positioned at an important entry point for how carbon enters soil environment, which is really critical for us to think about when we try and think of natural climate solutions, where we can try and build ecosystems that might suck more carbon and store it in a stable way below ground. Mycorrhizal fungi might really play an important role in those solutions. One of SPUN's recent research project was basically designed to count up how much carbon we're talking about here. How much carbon are plants sharing with fungi globally? And our estimates that we came up with were about 13 billion tons of equivalent CO2 is traded from plants to their mycorrhizal partners each year. Not all of that carbon stays below ground, but that's the sort of total bulk amount that we think plants are sending to their mycorrhizal partners. Now, to put that number in perspective, that's more than a third of the human energy-related emissions that happened last year. So that's a really substantial amount of carbon that's flowing from above ground into mycorrhizal hyphae. And we can start to look at looking at sort of like seasonal changes in plant growth and equating that to how much carbon might be flowing below ground in the maps on the right. So these are annual changes in plant growth, and then just scaled by how much carbon we think is sending below ground to a particular type of mycorrhizae. So not only are they really important for determining how carbon flows through ecosystems, they're an incredibly diverse group of organisms themselves on the planet. Some recent estimates suggest that there could be over 50,000 types of mycorrhizal fungi that form these types of mycorrhizal partnerships with plants. You can see a snapshot of this diversity probably in your local forays that you're doing, because they build really interesting and sometimes really delicious structures above ground as mushrooms. And this total diversity is much more than other types of organisms on the planet, like fish and birds and mammals, for instance. And while it's really easy and fun to go and explore the above ground structures that they make, I love to point out to people that they also make these really interesting structures below ground where they're interfacing with plant roots. So these images are either examples of the spores that they create to spread some of their genetic material, or the hyphal structures where as they're wrapping around or weaving their way inside plant root systems too. And I love these images because it just shows that, you know, it's not only the above ground world that matters. With a little bit extra work, you can go below ground and uncover lots of interesting details to these organisms too. And it's these points of contact along the root systems where the exchange of resources is happening, where they're sharing nutrients and gathering carbon. So with all of this diversity, how do we make sense of it all? And specifically with this large pool of diversity that seems incredibly important for our planetary health in general, where should we think about focusing our conservation efforts? And here we can use a framework like biodiversity hotspots. So these have been defined as places of really extremely high diversity of species, or maybe collections of really rare communities of species that in particular are facing environmental threats and potential extinction if we don't take some kind of protective action. UN scientists identified these about 36 different places around the world. And these hotspot maps have really been responsible for driving much of the global biodiversity conservation strategy of the last few decades. But what's critical is that they didn't include data on mycorrhizal fungi, really any fungi in general, when they were making these types of recommendations. And that's largely because they lacked the fungal data at the time to include in their assessments. And we think that's where SPUN can really step in. So we're working to build some of the largest and highest quality types of data sets of mycorrhizal fungi, so that these types of conversations or conservation strategists or conversations with legislators or land managers, anyone who's thinking about making a decision that impacts how well biodiversity might be protected in an area, has mycorrhizal information to bring to the table in a way that helps them make a more holistic decision about protecting biodiversity, not just above ground, but also below ground. In other words, we're trying to get mycorrhizal fungi a seat at the table here. And in order to do that, we need lots of data. So then the question becomes, how do we measure mycorrhizal fungi? How do we build these large data sets? We can do that in a couple different ways. We can look at above ground surveys of mushroom fruiting bodies, for instance, but not all mycorrhizal fungi make those types of structures. So it's hard to rely on those types of physical observations alone, although they can be really complementary. The approach that we're taking is a pretty common method in academic research these days, and that is sequencing DNA from the soil and then identifying the mycorrhizal fungi in those samples. So if you're familiar with environmental DNA methods, the same ideas apply here. Soil samples are collected in some kind of standardized way, and we extract the DNA from those samples, and then that DNA pool gets sequenced with fungal specific barcodes, and we can pull out the fungi strands within that large pool of DNA. And as you collect more and more samples, you can start measuring how different species are distributed among sites, and you can start calculating the total community diversity of a particular sample, for instance, by counting up how many different types of species are present. And now these steps provide a way to measure mycorrhizal fungi at a given site, which is really helpful if you want to identify which organisms are present there. But it can be difficult to then take that information you gain from sequencing one site and apply it to areas where you haven't sampled yet. So at SPUN, we've been tackling this challenge using a global data set of fungal DNA sequences to build predictive maps of mycorrhizal biodiversity. So this map shows the locations of all of the samples that have been compiled and processed together by our partner, academic partner, Global Fungi. And their team has encompassed and scraped more than 30,000 previously published sequencing samples that encompass nearly 3 billion unique strands of fungal DNA. And we're taking all of this data and we can calculate the total diversity of mycorrhizal fungi present in each of those samples. So that's what the color gradient shows here. Yellow is higher diversity that we calculate. And we can combine these diversity samples with many different environmental layers, spatial environmental layers, to train a machine learning geospatial model. The details of the model are a bit too technical to get into here. I'm happy to answer questions about it in the discussion section at the end. The main point is that we can train an algorithm to find relationships between fungal diversity and the particular environmental features of that sample where it was found, like the climate or the soil pH, et cetera. And then we can use those relationships to basically reverse engineer a map, to use those relationships to predict diversity in the spaces between these samples to get a full picture, or at least a full predictive picture of the current state of mycorrhizal diversity across the whole world. So this is an output from one of those model training data sets. So this map shows an output from the model. So each pixel here is a kilometer square resolution, and it is a prediction of how much mycorrhizal diversity we might find if you go and collect a given sample within that pixel. Bright yellow areas are places where we expect there to be the most species-rich communities of these network-forming fungi. And this map shows a particular type of mycorrhizal fungi, the ectomycorrhizal fungi, which are really common in temperate and boreal systems. So you see this bright spot of high diversity prediction across the sort of circumpolar boreal forest zone in high latitudes and some tundra ecosystems as well. But interestingly, there can also be pockets of them found in the southern hemisphere and tropical ecosystems as well, which our maps can also capture. But there are other types of mycorrhizal fungi. So this is another model that we ran for arbuscular mycorrhizal fungi. These fungi form associations with lots of grass species, some tropical tree species, as well as lots of crop species as well. And what you can see is that this is a fundamentally different diversity pattern than the other mycorrhizal type that I just showed you. So it's interesting that different mycorrhizal types are showing different diversity patterns, which is somewhat to be expected based on what we know about the types of plant species that they associate with. But again, this provides another data set we can share with folks who are making decisions. And we can say, hey, we have some information about how much diversity of different mycorrhizal types may be important in your area to think about from a conservation perspective. As we've started to analyze these maps, what's also becoming clear is that our picture of what makes a diverse ecosystem above ground doesn't always translate to below ground systems. And in general, there's a real disconnect between plant diversity and mycorrhizal diversity globally, which means that if we want to protect the world's most highly diverse ecosystems from top to bottom, we can't just rely on above ground indicators. So we think that's an important point to emphasize as well. And finally, we can actually look at how much mycorrhizal fungi are getting left behind in the global biodiversity protection strategies if we just isolate hotspots. So here I've just pulled out from those previous maps the most diverse areas that we're predicting in yellow, and also from some other separate modeling we've done, areas where we expect to find the rarest collections of mycorrhizal fungi in community together in purple here. So this is a mycorrhizal biodiversity hotspot map. What you can see are there are particular places where we expect high diversity and high rarity to occur, but what's great about this type of map is we can overlay other types of information, like where we currently have protected areas across the globe. So here are a couple zoom ins to different ecosystems where we've overlaid polygons that represent protected areas from a standardized database of protected areas. So these are national parks. These are state parks all the way down to regionally managed natural resource areas, anywhere that we're trying to limit human activities in some type of way. And as we run our analysis, we find that the total area of the hotspots here are well outside most protected areas as they currently are arranged on the landscape. More than 90% of the mycorrhizal biodiversity hotspots we predict fall outside protected areas. So it seems like that's more than 3 times more than other types of organisms like amphibians or mammals or birds, for instance. So mycorrhizal fungi have been systematically getting left out of where we're establishing protected areas globally. And again, SPUN is trying to get these data sets in front of as many people as possible so that they can start to be included as we think about protecting ecosystems. Now, I want to pause for a second here and remind everybody about a really simple idea, but a really important idea. And that's that maps can be incomplete and maps can be completely wrong. And there are 2 great examples of this. First is the Imago Mundi, which is largely considered to be humans' earliest sort of cartographic attempts. And this was humans who were representing Babylon at the center of this kind of circular world. And also even centuries later, Roman cartographers were kind of getting better at mapping the world, but clearly they were still struggling with this Europe and Asia and Africa map. So basically the idea is that just because you represent data on some kind of coordinate grid doesn't mean it actually reflects anything about the reality of nature that's happening in that location. And in our case, I'd like to think our mycorrhizal maps are not as wrong as these ones, but in many ways they are some of the first attempts at mapping in a globally coherent way the mycorrhizal diversity of Earth's ecosystems. And in our case, these maps come from statistical models, and that means 2 things. One, that they have uncertainty associated with them, but 2, we can measure and communicate that uncertainty when we're showing our maps of diversity. So the maps here on the left are 2 of uncertainty maps. Basically, the top one is how much variation there is when the model is making a prediction in a particular pixel, how much variation there is in its ability to accurately predict based on our validation test. And then the bottom map is an extrapolation. So how much the model has to guess basically about when it makes a prediction of biodiversity in a pixel, just by lacking how much it lacks information in the training data set that we supply it. So these are 2 really important maps to include when we talk about any of our biodiversity predictions, because as we include these, it will help decision makers have more of a full understanding of how confident we are in the prediction of biodiversity in that pixel, and so that we can try and maximize positive conservation outcomes and have an awareness of how wrong we might be in those predictions. But also what's great about working at SPUN is not only can we communicate the uncertainty in our models, but we can actually do something about it to try and reduce it. And this is a really exciting program that we launched shortly after SPUN launched, which is our Underground Explorers Grant Program. And this is a program where scientists from around the world can apply for funding to carry out soil fungal projects that specifically have a soil fungal DNA sequencing component to them. And so far, SPUN has granted a total of more than $750,000 to 92 researchers around the world. More than 80% of them are in countries in the global south. And the goal here is to try and accelerate global research by stitching together this network of soil fungal scientists around the world. And we're so thrilled about the success and the impact of this program, because it's allowing us not only to try and decentralize and democratize fungal science generally, but also it's able to gather data from really key places where we're lacking information on mycorrhizal fungi to then build into the next versions of our map so that they can become increasingly accurate. We're so thrilled about the success of this program and the response from the science community here. We're working really, really hard to keep this program going into the future. So now I want to switch gears a little bit again and talk about some of SPUN's new research priorities. And to do that, I want to bring us back to some old maps. So this is the Carta Marina from the 16th century, and it's regarded as the first detailed map of Nordic countries that included places and names. And again, I love this example as a way that maps can mix art and history and ecology together. But as you look closer, there's really one detail that sticks out, and it's the fact that there are monsters on this map, which feels very fitting for the season we all find ourselves in with Halloween approaching. The idea was that these cartographers included monsters or mythical creatures because they represented the edges of where our knowledge was at that time in history, places where we didn't have good detailed records of what the natural history was there. And so in reality, we may encounter sort of new beasts or new phenomenon that we didn't have descriptions for. And it's this type of idea that I want to bring us to, which is this question of where are the mycorrhizal monsters? What are the edges of our mycorrhizal understanding in this sort of mapping exercise that I'm walking you through? And we can think about two ways that we can define monsters. We can look at undersampled places, so places where we really don't know much in this data set we currently have about the mycorrhizal fungi in that region. And we can also look at unknown species, so places where there might be lots of undescribed, potentially new types of mycorrhizal species there with possibly unknown functions that could be really critical for those ecosystems. So we can look at this, we can dissect our model in a couple different ways. We can dissect our data set in a couple different ways and create outputs where we identify sampling priorities in blue and taxonomic priorities in red. So the sampling priorities come from our models that indicate where if we increase sampling efforts and sequencing efforts in these places, it could reveal new hotspots of mycorrhizal biodiversity that haven't been detected before. So these are extremely exciting areas to try and gather more mycorrhizal data from in the future. And taxonomic priority comes from an interesting work of fungal sequencing data. And that's the fact that in our data set, about 88% of the fungal sequences that we can definitely know are mycorrhizal, we can't assign a specific species name to. There's lots of reasons for this. There's a gap in how many fungal species, mycorrhizal species have been described from herbarium records, but haven't been sequenced yet, so that there isn't kind of a representative sequence of that named species yet. But what we can do is we can take the diversity of unknown taxa and build a map of them too, and identify places where if we sample, again, a lot of these places overlap with our sampling priorities. We sample these places and try and figure out which species are there, potentially are they new species to science. These are also really interesting research priority directions for us. And it's important to think about identifying new hotspots of mycorrhizal fungi or identifying new species because really a big arm of our ongoing and future research priorities at SPUN are thinking about threats to mycorrhizal fungi. So we think SPUN can really make a difference in understanding threats to fungi because we know human activities are causing harm to these organisms, whether it's sort of wildfire destruction and the loss of vegetation or certain agricultural practices that are slicing up these networks and really damaging them and only creating really intense selective pressures against their symbiotic effects. And it's easy to sort of ignore the destruction of these networks because they're underground, right? They're out of sight, they're out of mind, and their effects on ecosystems can be really hard to see and realize. So we've been trying to build data sets and spatial data sets so that we can start getting a better picture of where these mycorrhizal systems might be most under risk of extinction or at least loss of certain species. So this map is one example from some modeling work that a data scientist on our team, Clara Chin, is producing. And what she's been doing is she's been building lots of individual models of mycorrhizal fungi as they occur globally. This is for lots of ectomycorrhizal species specifically. And she's looking at essentially their vulnerability to abrupt changes in above ground biomass. So like deforestation events, for instance. And then you can compile all of those individual species models together to get a holistic community level assessment of where mycorrhizal fungi might be most at risk of going extinct or having lots of species decline from their communities if deforestation occurs. So the bright red areas here are places where we predict mycorrhizal fungi, ectomycorrhizal fungi in this case specifically, to be most sensitive to deforestation. Not surprisingly, a lot of the sort of temperate forest ecosystems and boroforest ecosystems are highlighted here, but you can see some subtle variation there. So it's not an equally consistent recommendation that we would make necessarily that all fungi are going to decline most severely by deforestation across all of the boroforests. We can specifically pinpoint where we think the most damaging effects might be. We can also look at other types of threats that mycorrhizal fungi face. So we can look at the distribution of fungi in a climatic sense. So this map shows all of the climate habitat that a particular Russula species is likely to occur in currently across North America. And what we can do is we can simulate future climates and remap the range of this species and examine how not only the species might shift its climatic suitability further north as climates start to shift, but you can also see where, like where Matt is living in kind of the Appalachian Mountains in the Southeast United States, there could be a real disruption in the climatic suitability for this species. And one research project we did was basically look at this climate disruption possibility for thousands of different forest tree and mycorrhizal pairs across North American forest. And we find that running these climate simulations shows that about a third of these mycorrhizal symbioses might be at risk of declining habitat, shared habitat suitability into the future. And we really don't know what those consequences might be if that amount of mycorrhizal symbioses are at risk. What does that mean for how nutrients get cycled in forests, or how healthy or productive those forests could become when they're facing new climates? And we can not only look at individual threats, but we know that threats compound and combine with one another on the landscape. So this is another project we have going where we're trying to synthesize all of these different environmental predictors that we know negatively affect mycorrhizal fungi to create kind of an integrated threat index. So we can combine different agricultural layers like fungicide, pesticide use, cropping intensity. We can look at climate layers like extreme heat events or drought. We can look at habitat loss, things like converting natural grasslands or natural forests to agricultural systems. We can look at soil pollution, things like compaction or mining areas or human density as a proxy for soil pollution. And we can weight these layers differently and combine them in different ways to create this combined threat index that we're starting to validate across other project areas that SPUN is working, like in wildfire affected forests in Italy and desertifying grasslands in Kazakhstan, places that in Lesotho are really struggling with soil erosion effects, and thinking about invasive coconut effects in Palmyra as well. And lastly, I think an interesting sort of future area that SPUN is thinking about is how these mycorrhizal systems might be useful to restore ecosystems that have already been degraded. And what's unique about this is that we're talking about restoring a symbiosis, not a single species, but really an interaction between plants above ground and fungi below ground. And I think corals are a really perfect analogy here. And I love how creative, all of these creative approaches that scientists are taking to rehabilitate and restore reefs, right? We've heard about coral bleaching and the destruction of coral reefs globally for a long time, it feels like. And I get really inspired thinking about people who are doing something about it. So this photo on the left is a coral reef nursery specifically designed for coral symbioses to thrive. And it's this type of ingenuity and real commitment to saving corals that makes me wonder things like, what would a mycorrhizal nursery look like? How would we build them? Where would they be? How big would they need to be to have, enough resources to rebuild mycorrhizal systems to promote the health of degraded ecosystems? And so if researchers can put on scuba gear and swim with sharks and make this happen for corals, why can't we be doing this for mycorrhizal systems, or at least exploring those types of possibilities? It's so amazing and really, really inspiring to me. So there's two things I want to leave you with today. The first is that mycorrhizal fungi are one of the most widespread and ecologically impactful forms of symbiosis on the planet. Historically, these fungi have not been considered when environmental decisions get made, but there's a growing community of scientists and policy strategists and advocates dedicated on changing that now. And the second thing I want to leave you with is that maps are important. They will continue to be powerful tools in how we make sense of the world, and also how we make decisions about protecting the planet's biodiversity. And at SPUN, we believe that mapping the underground, the diversity of mycorrhizal systems, can help correct some of the above ground bias that we've been suffering with in understanding and valuing the natural world. It can help establish a baseline for monitoring changes in mycorrhizal biodiversity, which is really critical. And it will improve our understanding of Earth's biodiversity and ecosystems and their functioning. So as a result, we believe it's really time to start mapping the underground with the same intensity and focus that we've shown we're totally capable of for doing for the above ground world. So that's all I prepped for you today. I'm really excited to get into the discussion with you all. And again, thanks for BMS and Lynne for the invitation to share some of this work with you all.
Thanks, Michael. Another absolutely superb talk. Maybe everybody could switch their cameras on and we could have a start on a general discussion for a little while. Excellent. And I thought perhaps we'd start with the very last question that's actually come into the chat, because it actually relates to all talks. It says, thanks all, amazing. And I certainly concur with that. And there's a general question: are you all biobanking, accessioning into museums, etc., your samples, soils, DNAs, etc., making these genetic resources available for future research because of climate change, blah, 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 and all those sorts of things? Maybe you would all like to answer that, perhaps Michael first.
Yeah, so we've been thinking about this for a couple different directions. So all of the samples that built maps that I was just showing are from published sequence sources that have been archived on places like NCBI, short read sequence archive. So they are open access and publicly available. It does take a little bit of academic or fungal specific knowledge in order to get some information out of them sometimes, but all of those samples are publicly available. In terms of all of the new samples that we're generating in partnership with our underground explorers, all of those, again, as part of the contractual agreement we have with those partners, are going to be open access and archived on those same short read archive databases as well. I mean, that's a core principle within SPUN, is to have data open access and available and interpretable by scientists and non-scientists alike. So definitely there are interesting wrinkles that happen when we're partnering with some researchers in some countries where there is a lot of sort of bioprospecting happening, and we're being really careful about making sure that they understand we're not trying to gather these data to build any type of proprietary product out of them or anything. And that with the new laws that are going in place in terms of shipping DNA around the world, all of those restrictions are becoming tighter. And so we're trying to do lots more sequencing in country when we're either in a place or working with a local community, asking research questions locally, which is another key mission element of SPUN as well.
Yes, thank you. Patricia, perhaps you could answer the same question.
Well, the answer is quite short. It's absolutely yes. I mean, all of the information we've been generating over the past years is publicly available on NCBI, and I'm convinced that as scientists, it's a very important part of our job to make the information available for the general public, right? I don't know you guys in the UK or the US, but here in Mexico, science is mostly funded by public money, coming from taxes from the people. So it's a main part of our job to make the information publicly available.
Absolutely. Did you want to add to that, Matt, or is that covered?
Sure. One thing I would add is that I think generations of scientists, there's been commitment from previous generations. I'm Gen X, and I think a lot of scientists in my career stage and age are very committed to open science, not only through depositing sequence data, but pre-printing things in bioarchive. It's this moral obligation to openness of data to accelerate solutions for a struggling planet. And whether it be information on DNA sequences on fungi or plants, I think all this data collectively can come up with solutions, but we can't do that if it's behind paywalls and only the select few companies are profiting from it and delaying progress. So I think that there's a real a sense of duty to make these things publicly available. I know, at least from my point of view, and the scientists that I consider colleagues and friends, they share those kind of thoughts.
Yeah, absolutely agree. And I guess that we also have a moral obligation really to make sure we write up as much of our scientific research as we possibly can for similar sorts of reasons, really. Bob, I don't know whether you want to add to that. It's not quite appropriate for your type of talk that you gave today, but if you wanted to add something, please
Yeah, well,
do.
it's certainly important that there are accessions in museums. And, there's so many fungal accessions that have been put into collections that have not been looked at or identified. And as I go through many of these natural history museums, it's amazing what's there, and no one has ever looked at it yet. And I think, again, as the museums get digitized, more and more of these examples are going to come to light, and we're going to find out a lot more about how they used fungi in the past.
Yeah. Thanks, Bob. As you're on screen, maybe I could ask some of the questions that were in the chat for you. I think particularly one was, it amuses me, this one. Were the noses on the masks glued on or part of the fruit body surface?
The masks from the Himalayas had noses that were attached, and they likely used some sort of tree resin to attach those. But they are the same fungus, and so they took the medicinal fungus, the medicinal Ganodermas, to make the mask out of. And not only the mask, but the attached noses too.
Yeah, and I don't know whether I half know the answer to this, but I've forgotten part of it. So I'm going to ask you and hope you can remember the rest of it. With regard to things like armillaria that luminesce, can you remember or explain the exact purpose of why the fungus is doing that?
Well, it's difficult to determine that. And, if the fruiting bodies glow at night, many people think, well, this may help in spore dispersal. Maybe this is attracting night insects, moths, and other things that would come and visit and move the spores around. But when you get to a fungus like armillaria, where the fruiting bodies are not glowing, it's the mycelium, and that's usually hidden, underneath bark, and you have to take that off to see it. In the Amazon, where we find mycena glowing, we can find fungal networks glowing under the leaf litter. And when you disrupt that, it starts to glow intensely. And so maybe in that case, the intensity in the increase after disruption would discourage predation on it and maybe inhibit, something from devouring it or whatever. So, it's difficult to say exactly why that these are producing the bioluminescence, but we have, many different examples where the fruiting bodies glow, spores glow, or the mycelial network glows.
I think they're all basidiomyces, and probably almost all, at least, wood decay basidiomyces. And I think that the reason that I read, at any rate, that's certainly to do with the mycelium, I don't know about the fruit bodies, is that obviously the wood decay fungi produce all sorts of nasty free radicals to decompose wood, and that hispidin is used to protect the fungus producing these compounds, and that within the cell walls of these fungi, hispidin is recycled, and that's where light is emitted. So it's a byproduct, suggested to be a byproduct of effectively protecting the fungus against the free radicals that it produces to do wood decay. So I do wonder whether it's perhaps, well, I don't know about the fruit bodies, but is that just an incidental thing that maybe invertebrates are attracted for dispersing spores? I don't know.
They could be, yeah. But also, some of the bioluminescence may just be from increased metabolism when something is disrupted and disturbed, and all sorts of enzymes get upregulated when that happens. And therefore, these other ones that are working on hispidin and other things are also accelerated.
Yeah. Thanks, Bob. Right, let's see what else. Let's turn to some questions that I've got here for Patricia. So some questions, these are questions from people from the chat. Are pharmaceutical companies involved in the discovery and development of medicinal compounds? or partnering with you and other researchers in this endeavor?
Yeah, well, pharmaceutical companies, not yet, but we are collaborating with an oil and gas company in terms of the bioremediation properties of the isolates that I just showed you. So they are very interested in these fungal properties because it seems that when they are drilling the oil from deep sea, the pipelines are stuck somehow because of cloaks. I don't know if it's the correct word in English, sorry. But it seems that they are interested in these organisms in order to allow the proper flow of the crude oil along the pipelines. So they are not interested in the bioremediation, but in some sense, they are interested in the fungal ability to use hydrocarbons. So I'm taking advantage of this interest to apply for bioremediation.
Yeah. Another question was, do any fungal species live on land and in the ocean? I think that's perhaps not talking about washing or whatever, but
yeah, it's a very good one. I'm actually very excited because there's this research group in India that are actually pioneers. It's a female leading scientist. She's a pioneer in deep sea research in terms of fungi. So there's this species, Aspergillus terreus. You know the specific epithet, terreus, means land in Latin. So this species is typical terrestrial Aspergillus. However, it's been repeatedly isolated and reported from the deep sea. So what the Indian research group experiments were to grow a fungal an isolate obtained from the deep sea of this species and grew it under deep sea hydrostatic pressure conditions and compared to a terrestrial isolate of the same species. And they observed very interesting differences in terms of growth under deep sea conditions. So hyphae seem to grow flat and wide. So they suspect that this may be an adaptation to the deep sea conditions in order to cover larger areas to obtain nutrients because deep sea is oligotrophic. And the terrestrial isolate failed to grow under deep sea conditions. So it's a small example, and this is a poorly investigated area, but I believe it's a very promising one.
Yeah, interesting. So, I mean, I think you've touched on something there. Of course, doing physiological or ecophysiological studies on these fungi. I mean, it's great that you have isolated fungi, and so you can do that on those that you manage to isolate. Presumably, there are very many that you have not been able to isolate in culture, and perhaps never, well, not in the near future, will be able to because of those conditions. So, I mean, I guess it's really hard to do the physiological studies because you need to have those conditions of pressure and etc.
Yeah, but fungi are not so susceptible, for instance, to the pressure differences. I mean, bacteria usually do not support. Deep sea bacteria are very difficult to isolate and grow in the laboratory, but fungi are somehow more resistant. So it's a pro of investigating deep sea fungi. Yeah.
Okay, good. Well, if we've got time, we'll come back to some more because I've got more questions lined up, but I must make sure I give everybody a bit of a fair crack of the whip. So I'll come back to you soon, hopefully, with more questions. So, Matt, some questions for you. I was amused by this one and rather liked it and feel the same. It's from somebody called Kate, and she says, what are the coolest entomopathogenic fungi we can find in the UK? And it says she got excited when she saw that they were prevalent on your maps.
That's a great question, and I'll start by saying that all entomopathogenic fungi are cool. That said, there are some cosmopolitan entomopathogens. When you think about Entomophthora musci, zombie fly fungus, a lot of these fungi, Tilipocladium, for example, These are things you can find in the UK. You can find in parts of Europe. You can find just about everywhere in the Northern Hemisphere and some in the Southern Hemisphere. What I would encourage those that are interested in is not only engage with your local mycology club, but use platforms like iNaturalist, where you could quickly ascertain the who's who in your local neighborhood. And oftentimes, if I'm looking for a specific fungus, the first thing I'll do is I'll go to iNaturalist and do a search for the US for that specific genus or species. So there are ways to do that. Again, it's only as powerful as people putting the data in, and there's some areas that are lacking in participation. But you can get out there and make observations and be the first to find Furia ithacensis, or the first to find, Entomoptera muskie. These are quite ubiquitous, or at least the species complexes are ubiquitous. Whether or not they actually are several cryptic species that require further scrutiny is not a question we need to ask and answer if we're just enthusiastic about finding things. But by contributing observations, we can then start to know the where of these fungi, like Michael was saying in his talk. I mean, the where is really the first part of it. And then the nuance of, well, we say it's where, but it turns out it's 3 species and they have subpopulations with some geographic overlap, but some with unique geographic locality data. So I would say check your iNaturalist page and look for observations, but you're going to find Bevaria, you're going to find Talipocladium, you're going to find cordyceps. Talk to Lynne. She knows a lot about these things. And talk to other members of the club, because there's a lot of knowledge there to really leverage.
Yeah, thanks, Matt. 2 questions then, which are of a similar form, but for different species. And the question is, any fungi known to affect pine bark beetles in Western USA?
So there's a lot we don't know, because there's a lot hidden in plain sight. Similar to what Michael talked about mapping the underground. I wish we could just look in the underground and see who's there, but we have to use high throughput sequencing to really ascertain the composition of those communities. Similarly, there's probably a lot of entomopathogens, more than we know, acting on a subset of bark and ambrosia beetles. But because they're under the bark or in the xylem, it's not really until we peel back the layers, literally, that we're able to see these mycost insects and try to start to understand how common they are, and the diversity of fungi contributing to those, those shortened lifespans. We only really trap and see a subset of all the insects that are in a given area, because only a subset make it all the way through the end of their life, because there are numbers of barriers to reaching adulthood and successfully mating. And some of those are fungi that are able to colonize those insects and take them out. The hope is that for the problematic beetles, that we could find candidates to leverage for biocontrol. But I would say that if we're seeing large beetle problems out west, and we have for a long time, and they're speaking to a larger problem. They're speaking to climate change. They're speaking to a change in fire regime. They're speaking to a change in age distribution and stem density across large swaths of geography. Until we address those things, we can't expect fungi to solve all our problems. They're only a tool to help us sustain our planet.
Yeah. I mean, you've sort of answered the next question in a sort of way already.
Sorry.
But no, that's absolutely fine, quite allowed. But the next one is specifically asking, any fungi known to affect guava moth in New Zealand? Because apparently guava moth is causing major problems with citrus crops and other things as it spreads downward in the North Island. I don't know if you know about that specifically.
Sure. Well, guava moth, I think, is what they call an owl moth. And there are fungi that infect that group of moths. I showed some pictures of myself looking quite surprised seeing a mycost moth infected with Acanthomyces. And Acanthomyces are known entomopathogens of moths. It's possible that some of these fungi can be leveraged against that pest. But, you know, before we go moving things around the planet, we have to understand, are there candidate fungi in that environment that could be mass produced, and then we could use a sort of inundatively as an inundative biocontrol before we start to move around things from location X to solve location-wise problem. But certainly I think that, you know, disease requires a coincidental overlap of a susceptible host, a virulent pathogen, a favorable environment. If you're saying there's a lot of suitable hosts there, then possibly a pathogen will emerge given enough the host, and we just have to be really on the lookout for it.
Yeah, thanks, Matt. I'll move on to asking Michael a few questions now. So one question in the chat is, how did you estimate the below ground fungal carbon biomass?
Yeah, good question. So that probably from the first part of the talk on carbon allocation. So, so we did a meta-analysis where we looked at all the scientific research that had been done that either traced carbon from plants to fungal hyphae in experimental systems. So you can label plants with a heavy form of CO2 that then you can measure in other parts in plant root systems, or you can collect hyphae and measure how much carbon is in that hyphae. And then you can look at basically what percentage of the amount of carbon that got labeled above ground ended up in fungi. So we collected all the studies that did that. And then there were also other studies that essentially measured fungal hyphal biomass in relation to plant productivity above ground. And we could relate the carbon flow basically as a ratio of plant above ground net primary productivity. And then we could crunch those numbers and come up with that kind of global number. There are other ways to do it more directly. If you measure hyphal biomass, you can then count up how much, you know, carbon molecules are in there in a number of different ways. But specifically, the main metric, the kind of gold standard, would be to somehow do some labeling above ground and follow where that carbon goes and measure how much ends up in the biomass of the fungi below ground. And of course, fungi are also being active and respiring CO2, and that's why I was careful to mention that not all of that carbon that gets sent to fungi actually stays below ground. They're using some of it to build their hyphal biomass structures, but they also create exudates like extracellular enzymes that go off and do different activities in the soil for them. And they're breathing out CO2. So some of that CO2 gets lost from the system as well. It's kind of a budgeting question, carbon budgeting question.
So perhaps following on from that, I could sneak in one of my own questions, and that is, without a shadow of a doubt, mycorrhizal fungi are hugely important. Nobody would dispute that at all. And mycological research really probably tends to focus perhaps on mycorrhizal fungi more than most other things in the natural environment. And you've just talked about carbon being shunted below ground, and also quite rightly mentioned that some of that is shunted back above ground by the mycorrhizal fungi. But I would like to know, is SPUN thinking about, or perhaps already doing this, looking at the saprotrophic fungi, the decomposer fungi, because probably they're as important as mycorrhizal fungi are. Their main job is sort of to do the opposite. It's the recycling part of the job, breaking down dead plant and fungal organic matter, and in so doing releasing nutrients. But simultaneously, of course, carbon is shunted back into the air as carbon dioxide. And of course, we talk about the Wood Wide Web and have done for a long time. I always think that there's more than this. That's usually meant to mean mycorrhizal fungi of the so-called Wood Wide Web. But to me, as a decomposition ecologist, I think there's more than one Wood Wide Web there. There's the decomposer Wood Wide Web. And sort of ironically, one of your pictures at the very beginning, where you were showing a corded mycelium, which I think actually is a wood decay fungus, not a mycorrhizal fungus there. So I don't know if you would like to comment on that. I'm thinking about, as a decomposer person, I'm thinking in the same way as you're thinking the mycorrhizal fungi have been left out of things. I think the decomposer fungi have been left out even more. maybe you'd like to make some comments on that.
Yeah, I mean, I would absolutely agree with you that decomposer fungi are definitely an important piece of the puzzle. We are not focused on them directly as an organization, but there are a number of our underground explorers who are doing local research projects in their areas where they are interested in the sort of interactions between symbiotic mycorrhizal fungi and the decomposer community to understand nutrient cycling or the effects on the ecosystem health in that region. But we currently don't have any projects where we're looking at sort of like the complementary diversity of saprotrophic fungi in relation to mycorrhizal fungi. I mean, as you know, it gets really complex the more folks you add into the party and trying to understand their interactions. As we start to explore more questions and research projects related to mycorrhizae and carbon, though, I do think we're going to have to include more some more context, more information about potentially the decomposer community there to understand more of that carbon budget that I was mentioning.
Absolutely. Perhaps I could just ask you one more before returning to a couple of the other speakers. And I promise I will try to wind down this discussion before too long, because I think you guys have been really grilled on this with all sorts of complicated questions. And great as the discussion is, I really don't want to sort of overtax you. That's got not, take you too much for granted, let's say, put it that way. Anyway, another one for you here, Michael. You explained very nicely about how above ground plant diversity really doesn't correlate well with below ground fungal diversity. But the question here that was put in the chat is, which above ground features show the strongest correlation with fungal diversity? And have you managed to look at that? For example, do you look at geology or anything like that?
There are some geology layers. We have like hundreds of global spatial layers that go into this model, and there are some that reflect geology, like distance to parent material, underlying soil type, those types of more geological features. Some of the key variables that come out are actually different depending on which mycorrhizal type we're modeling. For ectomycorrhizal fungi, it's above ground biomass is the strongest predictor of the most biodiverse hotspots of ECM. So basically, forests that are really large, like old growth forests, basically come out as a big predictor of ectomycorrhizal fungi, which is really exciting in a number of ways, because in a complementary way, our above ground remote sensing is accelerating so fast to the degree that we can isolate old growth forest structures using remote sensing tools like satellites and whatnot. So if that's the main predictor of ectomycorrhizal diversity, that might become kind of like a bridge for us to identify regions where there could be hotspots of ECM diversity. For arbuscular mycorrhizal fungi, it's a host of other types of variables that are the strongest predictors, like temperature, some sort of strange soil variables that are kind of surprising to us, as well as there's a signal of sort of human modification of the landscape, basically. And we think that could be because as you either disturb ecosystems, you might have more non-native or invasive plant species in an area, and that might kind of artificially inflate the diversity of arbuscular mycorrhizal fungi present in those samples. But yeah, definitely an intriguing signal to kind of explore further.
Thank you, Michael. Patricia, I love the fact that that bacteria hitchhike on fungal mycelia from the ocean depth, and of course we know that occurs in terrestrial ecosystems as well. So you have them in that wonderful maze layout. So what do you think happens when? So presumably they must swim along the surface of hyphae, because you can't sit on the hypha and get there. Hyphae don't work like that. But what happens when they swim along to the tips of the hyphae? Any thoughts of what they do when they get to the end? Where are they going? What are they trying to do? What are they trying to get to?
Well, first of all, we observed that when the fungal isolate reached the end of the maze, somehow bacteria detected some signal. We're not sure what kind of signal, but in this part of the isolate, the bacteria colonized the agar medium. That's why I used the expression, they jumped off the hyphae.
Okay.
And also we observed that in some regions of the fungal isolate, bacteria altered the growth of the fungus in a negative way. So this confirmed the hypothesis that this cross-kingdom interaction is perhaps mycophagy.
Interesting. Yeah, just to, I shouldn't really be putting any of my own work forward, but I'm going to, because this is quite interesting how this sort of fits in. When we've looked at bacteria which track along mycelia and mycelial cords in soil, it's quite interesting that sometimes when we do lots of these pairings with one fungus and another fungus to see which one beats each other in these pitched battles. And sometimes the presence of a bacterium can make a fungus more aggressive, and sometimes it can make it least aggressive. So there are both positive and negative effects.
Yeah, well, actually, there's some information I did not include in the presentation because of time. We are now working with the experimental evolution of this cross-kingdom interaction. So we are tracking the genomic changes across several generations of these two organisms, the fungus and the bacteria, when co-cultured and comparing with the monocultures. So it seems in terms of, for example, ascomata formation, we're not sure if it's the result of the interaction with the bacterium, but in the co-culture experiments, after several generations, the fungus stops producing ascomata. So it's very interesting. Yeah, we need to go deep into the genomic data to explain this pattern, but this is ongoing work.
Just one more quick one on this same topic. Do you find that bacteria are only specialize in maybe or only can track along certain fungal species?
I'm not sure about that. It's a tricky question because we are working only with a strain of Bacillus. Yeah.
Okay, yeah.
And this strain was not the dominant ASB from the metabarcoding of the 16S gene. So I'm not sure about that.
Yeah, fair enough. I was wondering, do you have any idea? Well, you must have some idea. What proportion of the fungi that you managed to culture? What proportion of all the fungi that you know there from your non-culture techniques as well are actually culturable? Is it just a very small, very tiny proportion, presumably?
Yeah, very small proportion, but interestingly, it's complementary to the high throughput ITS dataset. So we have 2 or 3 OTUs that were not as good, as well represented in the culture independent dataset. So I'm sure that it's very important to use a complementary approach when describing fungal diversity.
Yeah, absolutely.
Lynne, if you don't mind, I'd like to chime in on something that Michael said, but it relates to what Patricia was also saying, is that
please do, yes.
Recently I was working on a project that involved fungivorous millipedes, and it's unrelated necessarily to this topic, but it speaks to a larger problem. We first did a culture-based study, and we were trying to assign guild or niche to these different fungal taxa from the culture-dependent study. And, we were able to assign, say, 80 to 90% of them to a guild using funguild or fungal traits. When we look at OTUs from the same millipedes using a culture-free or DNA-specific approach, we're only able to assign less than 30% of them to guild. I think this speaks to the problem about decomposers. There's a lot of fungi that are specifically in groups, polyporales and wood decay fungi, that we know that they're capable of serving as decay fungi. There are soft rot fungi, which are scattered among fungi, which we know that some can cause soft rot. But I think until we get a clearer picture of the different ecological niches that these fungi fill, it's a lot, there's a lot more barriers to mapping them and understanding how they're interacting, because we still can't even assign a function, like not even a facultative secondary lifestyle. We can't even assign a primary lifestyle to many of these fungi. And until we solve that problem, it's hard to move forward and do the kind of decomposer mapping I think that you're talking about.
Mm-hmm. That's a very, very good point, Matt. And I think sort of allied to that also is wrong categorizing of what the roles of fungi are. I mean, to me, the classic example is in most of these databases, if you find a fungus, or a wood decay fungus, say, which is fruiting on a tree, it is often classified as being a pathogen of the tree. And more often than that, that's very far from the truth. It's probably a heart rot fungus, and of course the heart that colonizing the heartwood in the center of the tree, which is dead, and so it's not a pathogen because you can't kill something that's dead, and it maybe does not invade living tissues. So it's, we've often got things wrong as well when we try to categorize things. So yeah, good points. Good points. Well, while we've got you there, Matt, let's just ask you another quick one. We'll stop in 2 or 3 minutes because we've nearly had 40 minutes of discussion. Yes, I think you mentioned that the huge potential as biological control agents for these insect pathogenic fungi. And we know how important potentially using them as biological control agents rather than using chemical pesticides might be. And we know that people have worked on this in the past, but biological control on the whole hasn't really yet taken off, has it? Do you think that it will? And do you want to comment upon the reasons?
Sure. There's been a lot of examples of biocontrol failures, and it's not for lack of trying. In fact, there was a paper put out, I feel like 15 years ago, talking about the amount of time investment and money to bring a biocontrol to market, over a decade of research and X number of dollars with like no guarantee of success. No company wants to invest in something that's not going to work. And oftentimes we have generalist entomopathogens, which, could jump to our beneficial, predatory insects that we're using to manage. So, finding host specialists, much like we use rust fungi as biocontrols of plants because they tend to be host specific, is useful. But oftentimes, more than not, these fungi are biotrophs, obligate biotrophs, and we don't really fully understand their life cycle because we can't culture them away from their hosts. A lot of the fungi that I talked about today in the Entomopterales, Mesospora, Cicadina being the primary one, we've never successfully cultured it, nor has anyone else. I'm not saying that we're incapable of that, because people can lean on the, Xylella fastidiosa as a fastidious prokaryote that we couldn't culture for 100 years until we figured it out. But if we invest so much time in figuring it out, no one's going to fund that. And two, it just prevents these huge barriers to using these things, because you need a host to generate inoculum, much like you need to do a generation of AMF spores. You need to culture them on Sudan grass or whatnot, and then mass produce them and then use that as inoculum. So there's these challenges that you have to kind of rear them alongside or on their hosts. And we can't fully decouple host responses from pathogen, from responses without being able to purify and culture these organisms in the lab.
Yeah, absolutely. Yeah, thanks for that, Matt. I think one last question for Bob. We haven't asked you so many questions, Bob. I was wondering where to now, and perhaps before we get to where to now, I wonder how exactly did you get into doing all this wonderful ethnomycology? were you invited by various places to go and do various researches? For example, Ahmed earlier talked about you going over and helping with this with stuff in Egypt, or was it a research inquiry that you initiated yourself and got hooked on, and you know?
Yeah, well, I'm invited to go to work on other projects usually, and they involve wood decay and degradation of some of the objects, and taking a look at the organisms, whether they're in the desert of Egypt or in totem poles in the Pacific Northwest. And there, working on those, you know, I was able to find information about the fungal objects in the various museums. And then finding one leads to another and another. And there are just so many that are in these various natural history museums that haven't been looked at or evaluated or information about. And one of the real interesting things of this is that there's a lot of collection notes present for them. So you get, you know, information that is real. You know, often when you're talking about historic uses, you know, you need to have some knowledge about, well, is this really true? And, you know, in those museums, you've got those collection notes made at the time that the fungi were collected and put into the museum. And it's just, I think, unfortunate that a lot of these were just sort of left in the collections and nothing was done with them. But I think now more and more can be, and we can find out a really great deal about how these were used by various shaman and others in the past. And of course, remember that, you know, these fungi were collected back in the 1800s before people had a very good understanding of what disease was, you know, and what was causing it. And through trial and error, they were able to find fungi that worked. And we find today many of these that we know have medicinal properties were the ones that they used.
Yeah, yes, it's absolutely fascinating, isn't it? Yeah. Thank you very much, Bob. Okay, so last question, and this one is to Michael, and it's from Charlotte. And she says, in school, it's taught that nutrient mineral leaching in soil can be prevented by the presence of plant roots. Could I assume that mycelium networks work in a similar way to prevent leaching of carbon? Well, it says carbon from the soil, but perhaps perhaps it means mineral nutrients like nitrogen, phosphorus, and things like that.
Definitely mineral nutrients, yes. I mean, the mycorrhizal fungi in general are pretty well thought to, you know, increase the functional surface area of plant roots. So they do definitely help reduce leaching. So the presence of plant roots probably that you're learning about are effects of mycorrhizal fungi that are preventing the leaching of nutrients because they're able to gobble them up before they leave the system. Leaching of carbon is an interesting question. I'm thinking of like maybe bicarbonate leaching into rivers or something like that. There are definitely at SPUN, we're interested in thinking about fungal, like mycorrhizal fungal biomass as a way to potentially help increase the carbon drawdown capabilities of ecosystems. But again, we have to be really careful about that because not all carbon is going to stay below ground, and we don't know how, if we increase the abundance or diversity or biomass of mycorrhizal fungi, what is that going to do for the decomposer community, like Lynne was mentioning, and will that actually serve as a priming effect where they might start mining, you know, organic matter and upregulate their CO2 emissions, and so there might actually be like a loss of CO2 from the system. So the leaching of carbon, or in general, the carbon budget as it relates to mycorrhizal fungi is pretty, it gets complicated really quickly. But certainly for nutrients, mycorrhizal fungi are a great way to reduce the leaching of nutrients.
Yeah, thank you. I think at that point we should probably stop. I'm sure we could go on for quite a lot longer, but I expect you're all getting pretty tired now after giving wonderful presentations, and I'm not just saying that, I truly mean that. And indeed, the chat indicated that there was huge enjoyment from the audience, and lots of thanks from people. Also, great discussion, everybody. So thank you so much. It's been a wonderful session. So thank you to all of the speakers for today, for the whole of the day, and particularly obviously at the moment in this session. And thanks to Sally for making sure that everything ran so smoothly. And hope to see you again soon. And let's hope we have another symposium like this time next year. So thank you very much, everybody. Have a good evening.
Thanks, Lynn.
Thanks,
Bye.
Lynn. Thanks so much.
Thank you. Bye.
Thank you and happy Fungus Day.